Hello students, how are you all? Stay safe. Already we saw in live class how do organisms reproduce. Organisms reproduce by asexual and sexual reproduction. Now we are going to see part 1 under that heading subtopics introduction, types of reproduction, asexual and sexual reproduction differences. Then types of asexual reproduction, advantages of vegetative propagation. Introduction. As you all know, reproduction is very important because the continuity of life depends on reproduction. Reproduction is a biological process in which an organism produces their own kind or offspring. In this process, there is creation of a DNA copy. It ensures the continuity of life. It helps in increasing the species of same kind. Then, differences between asexual and sexual reproduction. Asexual reproduction means only one parent is involved, no involvement of sex, no reproductive cells or gametes, no fusion and fertilization, no variation is also occur. Sexual reproduction, two parents of opposite sexes are involved. One should be male, one should be female. Production of sex cells or reproductive cells, or we can say gametes. They are fused and that process is fertilization. So fusion and fertilization takes place. Then lot of variation is seen. Asexual reproduction. What is it? It is a type of reproduction in which a new organism arises from a single parent without the involvement of sex cells or gametes. Types of asexual reproduction. First one, fission. Fission means break. Binary fission. So divided into two. Best example, amoeba. Multiple fission. Plasmodium, best example. See here in this picture, you can see how the amoeba is. Yes, you can see here the amoeba. First divided, the nucleus is divided into two, that is followed by cytoplasm. So two individuals are forming. Fission. Fission. This method of asexual reproduction takes place in unicellular organisms like amoeba, paramecium, bacteria. In this case, the parent organism divides into two daughter cells. Here first the division of nucleus takes place, that is called karyokinesis. Then division of cytoplasm, that is called cytokinesis. Yes, binary fission. Here you can see that amoeba, here this amoeba, the nucleus divides into two. Then that is followed by cytoplasmic division. Then we are getting two offsprings. So two new organisms formed from a single individual. Best example, amoeba, paramecium. The parent organism divides into many new organisms at the same time. That is only multiple fission. During unfavorable situation, the organism develops a protective outer covering called cyst around it. Inside the cyst, the nucleus divides into many pieces. Okay, each nucleus takes little bit of cytoplasm and forms a large number of daughter cells. This is surrounded by a protective layer called cyst. On the onset of favorable conditions, the cyst wall rupture and daughter cells are released from the cyst. Okay, so new organisms are formed. Best example, Plasmodium, 1000 cells are produced at a time. Budding. In budding, a bulging on the body appears. Okay, bulging of body. See here, you can see here the bulging. This is only bud. Then it undergoes repeated mitotic division of cells here. Therefore, this lateral bulge is called bud. The bud further grows and develops into a new organism. Okay, here. Then it forms a mouth and tentacle. Then after few days, detached from the mother and finally into a new individual. Okay, best example, Hydra for budding. Yes, this is budding in yeast. Chain of buds are formed. Okay, spore formation. In fungus, a structure called sporangium develops from hyphae. Hyphae is nothing but plant body. Here the nucleus divides and redivides. Each nucleus takes little bit of cytoplasm and develops into a spore. When the sporangium gets filled with the spores exerts pressure on the wall of the sporangium. Yes, as a result of which the wall of sporangium rupture and spores come out and germinate into a new hypha after getting suitable substratum. Already we saw in the online class also. See here, this is a sporangium we can say. This sporangium, inside that spores are found. Okay, then it rupture and spores are released. So it gives new organisms. Okay, best example, already we saw bread mold or rhizopus. Regeneration. It is the ability of an organism to replace its last body parts or full organism. In higher organisms, only few parts can regenerate, but lower organisms, the full body. Example, Hydra, Planaria, Sponges, Spirogyra. Spirogyra. The process of regeneration is called a fragmentation. As the mature Spirogyra breaks into two or more pieces and grows into new individual. Even we saw lizard is also having regeneration capacity. Its tail. If it see the enemy, immediately leaves its tail and later it develops. Even human also has some regeneration capacity. Nail, hair, skin. Yes, in this diagram you can see how the regeneration occur in Hydra. See here, in Hydra, it is divided, the upper part, lower part. The upper part can produce the lower part can produce the upper part. Okay, here this is a planaria divided into three portions. So the upper can produce middle and lower, the middle can produce upper and lower, the lower part or bottom part can produce middle and upper. So best examples are Hydra, Planaria. Fragmentation. It takes place in some multicellular organism. Best example, Spirogyra. Yes, you know it is a pond silk. The organism breaks up into smaller pieces upon maturation. These pieces of fragment grow into new individual. Here two, then this part can produce the remaining, this part can produce the upper half. Like that way it can produce. Vegetative reproduction. It takes place in higher group of plants. It is also called vegetative propagation. In this case, new plants are obtained from the vegetative parts like roots, stem, leaves, branches of the plant. Example, rose, jasmine, sugarcane, bryophyllum, guava, potato, onion, mint, strawberry, lily, etc. We can say otherwise the plants which cannot produce the seeds, they can grow by their vegetative parts. In bryophyllum, you can see the leaf margin can give new individual, okay, leaf margin. Then here tissue culture, from a piece of tissue, new plants can grow in the nutritive medium, okay. Then here you can see the cutting, best example you know very well, drumstick we can say. Then layering, jasmine or rose plant, even rose plant we can say cutting also, layering is also possible. Then crafting, already in the online class also we studied about this. This is a stalk, this is a scion. Then they are fixed with the help of a polythene cover with the soil, okay, without damaging the tissues. Then this is air layering, okay. These are the methods are called what? Artificial vegetative propagation. Types of vegetative reproduction. First one, cutting. A cutting may be a piece of stem, root, leaf or a bulb scale. Okay, a part of the plant which is removed by cutting is called cutting. In this process, a cutting of the plant is partly under moist soil, as a result of which root and shoot develops and it behaves as new plants. Rose, sugarcane, chrysanthemum. Yes, here it is shown, rose. Cutting, then they are making it in other part, then it can grow as a new plant. Layering. In layering, a branch of the plant is pulled towards the ground and a part of it is covered with moist soil while it is attached and supported by the parent plant. You can see very well here, this plant is here, the last part of the portion is fixed inside the soil. It is supported by a parent plant. After few days or after some time, new roots are developing. Then even leaflets also we can see from that. Then it will be removed from the parent by a cut. Okay, then the branch is cut off from the parent plant which behaves as a new plant. Here the new plant is shown. Grafting is a new method in which stems of two different plants are joined together in such a way that they will unite and grow as one plant. In this diagram you can see very well the portion which is removed, okay, the plant which is in the soil is stalk, removed portion that is scion. It is attached to the stalk. Okay, the portion of a plant that is grafted onto other plant is called scion, and the plant in which grafting is done is called stalk. By doing this process, the cambium of scion and stalk come in contact with each other. We can get different varieties by this process. Example, lemon, orange, grapes, rose, pineapple, etc. Advantage of vegetative propagation. The new plant is similar to the parent plant, so desirable characters may be replicated. The new plant is identical to the parent, no variation. Okay, we can reduce the period of maturity, flowering and fruiting. Within less time, within less cost, we can get more profit also. We can generate different varieties. Tissue culture. It is called a micropropagation. Tissue culture is a process by which new plants are obtained from small pieces of plant tissue. And culture medium. Culture medium contain nutrients and hormones for the growth of the tissues. Example, banana, bamboo, orchids, dahlia, carnation, etc. Advantage of tissue culture. We can develop disease-resistant variety, high-yielding variety can be obtained. We can develop early flowering and early maturing. We can develop haploid plant. The process of development of a plant from an unfertilized egg is called parthenogenesis. It is helpful in developing seedless fruit, for example, banana, grapes, orange, etc.